Hello, my name is Mandy Frazier, and I'm a PhD candidate at UC Davis in Ann Todgham's lab. My work centers on Antarctic fish physiological ecology, and I'm especially interested in understanding how Antarctic fishes may cope with changing oceans. Fishes play an important role in the Antarctic marine ecosystem, and today I'm going to tell you a brief story of how two of the most common notothenioids, Trematomus bernacchii and Trematomus pennellii, may cope with climate change. As you are all aware, climate models predict continued rapid change in the Antarctic, including warming, acidification, ice melt, and freshening. With these environmental shifts, we anticipate drastic changes to the ecosystem, such as invasions of more temperate species, range contractions, and the complete loss of certain vulnerable species. I use a comparative framework that examines interspecific sensitivities to climate change to help understand how ecosystems may restructure in future oceans. Now, likely due to their higher abundance, bernacchii have been studied in much greater detail than other benthic trematomids, and they have been treated as the representative generalized Trematomus species. But there is some evidence that bernacchii may not actually be representative of other benthic notothenioids in their physiological capacities to cope with stressors. In fact, we think that pennellii, as somewhat overlooked and very closely related species, may be better prepared for climate change than bernacchii. There's growing physiological evidence that supports this hypothesis. In a heat tolerance study, pennellii survive significantly longer in extreme heat exposure, 14 degrees Celsius, both with and without a previous warming acclimation to 4 degrees Celsius. In a cellular energy budget study, pennellii hepatocytes, or liver cells, were able to ramp up their cellular respiration at high temperatures without any change in energy budget, while bernacchii hepatocytes showed limited metabolic capacity at these high temperatures. In our group, we have ongoing studies showing that pennellii may have greater onboard energy stores on the organismal level, as measured by Fulton's condition factor. We also have data indicating pennellii have significantly lower resting energy requirements, as measured by their resting metabolic rate, but that they have higher mitochondrial respiration capacity. Together, these data indicate that pennellii may have much greater energetic capacity to cope with the stressors of a changing climate. There is also ecological evidence suggesting that pennellii may have a stronger ability to cope with future oceans. A diet study examining gut contents found that pennellii had a more varied diet than bernacchii, and a more generalist diet may be adaptive if other ecosystem shifts occur. In another study, pennellii heavily dominated areas disturbed by iceberg scouring, suggesting that they may be better able to recover from habitat disturbances. Notably, and finally, preliminary data from our group indicate that juvenile pennellii may actually prefer warmer water than bernacchii. As you can see, the comparative data that we do have supports this idea that pennellii may actually be better suited to cope with future oceans. Species-specific differences in vulnerability to ocean change could drastically shift the distribution and the abundance of benthic fishes, particularly if bernacchii, the more abundant fish, are more vulnerable. As oceans continue to change with climate change, ecosystems are predicted to restructure, and some species will be winners, and some will be losers. Benthic Antarctic fishes play an especially important stabilizing role in the Antarctic food web, and thus may amplify climate change-related perturbations throughout this larger food web. Understanding the capacities of benthic notothenioids therefore informs our understanding of impacts on the larger ecosystem. With that, I'd like to thank you for watching this flash talk, and I'd like to thank NSF and UC Davis for research funding. And I'd especially like to thank my team and all of the McMurdo staff for making this research possible. If you'd like to get in contact with me, my Twitter handle and my email are both on the slide. Thank you so much.